A population is in Hardy-Weinberg equilibrium if its allele and genotypic frequencies remain at equilibrium after one generation of random mating. There must also be an infinitely large population, no selection, no migration, and no mutation. A rare species of penguin found only in Antarctica has a specific gene that controls feather pigment. It can have either the A allele or the B allele. Three different phenotypes for feather pigment are found in these penguins. The first is a purple underside, the second is blue, and the third is white. Before selection occurred in a population of 200 penguins in Antarctica, 46 of them were purple, 100 were blue, and 54 were white. Given the number of individuals for each genotype, we can calculate both the allele and genotypic frequencies to determine if the population started in Hardy-Weinberg equilibrium. To find the frequency of the A allele, we divide the number of A alleles found in the population by the total number of alleles. The frequency we get is 0.48, or the p-value. We make the same type of calculation for the B allele, which gives us a frequency of 0.52, or the q-value. To find the genotypic frequencies, we simply divide the number of individuals for each genotype by the total number of individuals, giving us these frequencies. Now that we have found the P and Q values, we can use a Punnett square and the bolded formula to determine the genotypic frequencies in future populations if they remain in Hardy-Weinberg equilibrium. The frequencies found align with the number of individuals in our penguin population, confirming that it is in Hardy-Weinberg equilibrium, provided that the previous assumptions have been met. Over many generations, selection occurred in this population of penguins. There are now 31 purple penguins, 95 blue penguins, and 74 white penguins out of the 200 observed. To determine the new genotypic frequencies of this population, we multiply our initial frequencies by the fitness of each genotype, which tells us how likely individuals are to survive to reproduction. The unadjusted survivor's value is divided by the sum of all three survivor values to give the scaled genotypic frequencies of the population after selection. We can multiply these frequencies by the total number of individuals in the population to see how many penguins have each phenotype and genotype. Notice how the number of purple and blue individuals decreased after selection, while the number of white individuals increased. The changes in the number of individuals with each phenotype demonstrate directional selection. Penguins with a white underside are favored over penguins with purple or blue undersides. The population is no longer in Hardy-Weinberg equilibrium since one phenotypic extreme has been favored over the other through selection. The white feather phenotype is favored over the other phenotypes because white penguins can easily hide from predators. When they are in the ocean, their white undersides blend in with the sunlight shining through the water, protecting them from predators like the killer whale. In future populations of this rare penguin species, we can expect to see the number of white penguins increase and the number of purple and blue penguins to decrease. Due to the white penguin's advantage over predators, they are favored in selection to ensure the survival and reproduction of the species, violating Hardy-Weinberg equilibrium.